This is a video for topic 1.6 in the IB Biology course for the topic of cell division. So once again, here are all the understandings, applications and skills over here for this topic. It's very important to know all of them. This topic is obviously about cell division, which starts with the cell cycle. So the cell cycle is composed of 4 stages, or in 2 stages in a way, but one stage is separated into 3. So as you can see, there is interphase, which is the stage of development of a cell between 2 successive divisions, and that interphase is separated into G1, S and G2. G1 being where the cell grows, prepares for replication, S is the synthesis stage, and G2 is more growing and preparing for cell division for the cell. The M phase, or mitosis, is the period of the cell cycle where the cell and its contents divide. So mitosis is also separated into multiple areas of prophase, metaphase, anaphase, telophase, and cytokinesis, which is separate from those other 4. So there's the sort of functions that occur in interphase, so normal cell functions occurring. DNA supercoiling is how DNA is packed tightly, and it needs to be unpacked for replication to occur. So chromatin is the DNA loosely packed within the nucleus, and it's very accessible for transcription. A chromosome is a tightly packed and tightly wound DNA, very condensed, so to be much more efficient for mitosis. A chromosome is the condensed form of the DNA, which is visible during mitosis. As shown here, so this is the chromosome, and then it replicates, causing sister chromatids, which are the 2 sides to this, and then daughter chromosomes once again. During mitosis, the 4 phases that we spoke about earlier, prophase, metaphase, anaphase, telophase, are shown here. As you can see, prophase is the DNA supercoiling and the paired centrosomes, these little bits here, moving to the opposite sides of the cell and forming microtubule spindle fibers, which are these hair-like projections. On top of that, the nuclear membrane breaks down. In metaphase, the microtubule spindle fibers attach to the centromere of the chromosome, which is this small bit connecting the sister chromatids. And in anaphase, the continued contraction of the spindle fibers causes each sister chromatid to pull apart. Once they're separate, they're considered their own chromosome. Telophase is when the chromosomes arrive at either side of the cell, and the cell starts beginning to change shape into 2 separate cells with nuclear membrane reforming. Cytokinesis is the next stage, and is the separation of the cells. In animal cells, it's caused by a cleavage furrow and contractile rings that constrict the joint between the two sides of the cell, eventually cutting them off, and 2 separate membranes are formed for 2 separate cells. In plant cells, however, due to the existence of the cell wall, a sort of cell wall begins forming in between the 2 sides of the cell, called a cell plate. This fuses with the cell wall and separates the cytoplasm. Once you've reached this stage, the 2 sides can pull apart as they are separate. Another thing about mitosis, a skill that you need to know, is how to calculate mitotic index from looking at microscope images. So here we can see heaps of cells with many undergoing mitosis. So the ones you can see as mitosis are the green ones here, and they're ones that resemble one of these stages. So to calculate mitotic index, you count the number of cells that you can spot in the process of mitosis over the ones without, that aren't in mitosis. So no visible chromosomes in here, just the nucleus. The cell cycle is regulated by cyclins and cyclin-dependent kinases. And so the way they work here is there is a cyclin and a cyclin-dependent kinase that bond, and once they're bonded, they are in their active forms, and they can cause this expression of different things, causing certain functions to occur. As you can see here, cyclin A goes from S to G2 phase, cyclin B goes from G2 to M, cyclin D from M to G1, and cyclin E from G1 to S. So that's when they're most active at that certain point in the cell cycle, and so they're responsible for moving the cell on when it's the right time for it. On top of mitosis, you can have tumours, so that's abnormal cell growth resulting from uncontrolled cell division. That can occur in any tissue or organ. A mutagen is an agent that changes the genetic material of an organism. Mutagens can be physical, chemical, or biological. Here's some sort of examples of things that could cause mutations, and mutations that cause cancer are classified as carcinogens. Oncogene is a gene that has the potential to cause cancer. So most cancers are caused by mutations of two basic types of genes. And so here, proto-oncogenes code for proteins that stimulate the cell cycle and promote cell growth and proliferation, and tumor suppressor genes code for proteins that repress cell cycle progression and promote apoptosis. Metastasis is when cells can spread from, so tumor cells can spread from their original location. So, for example, having lung cancer that then spreads into another area of the body or the organism. So the original location is classified as a primary tumour, and other locations that it spreads to are classified as secondary tumours. There's been heaps of scientific studies into the link between smoking and cancer, and as you can see from these graphs here, is very convincing evidence. As there's over 60 chemicals in a cigarette that are known to be carcinogenic, very strong correlation, and smoking has been linked to dozens of cancers and increases the risk significantly. There you go, that is everything that you need to know for cell division and topic 1.6.